Right. Good afternoon, everybody. Thank you for being here, particularly Ada and people of all ages, as well as to the folks joining us online. We have the distinct privilege of having with us today Professor, I should say, Greg Appelbaum, who's joining us from the University of California, San Diego. He is in the Department of Psychiatry there. But before getting there, he's ping-ponged back and forth across the United States from coast to coast, training originally in North Carolina at Duke, if I remember right. California at Duke. Yeah, sorry. I got it all wrong. This is why I should not introduce people. Ping-ponging between the two, but always ping-ponging with a vision. And among his amazing research are a few items that I think are worthwhile, which I think you'll talk more about. But Greg is an expert not only in vision sciences, but also in bringing vision to life and showing how we can take what we understand about sensory processes and movement and putting that into daily practice. And I think that's one of the real achievements you've already attained, and where you're also continuing to make absolute headway in terms of bringing neuroscience into daily life. And that can be by combining behavior, combining interventions, combining measurement, working with artists of various sorts, athletes of various sorts. And I think it's exemplary, really, of where neuroscience needs to go. And for that reason, I'm particularly pleased that you're here. And on top of it, I'll make a little pitch for Louise, who is a student with Greg, who obtained a fellowship to come here for a short stint. But this exchange between our labs and the sense, I think would be something to certainly foster and hopefully get you here for a bit longer than a few days. So we're profiting from the fact that you were in Helsinki for a conference, and now we get to enjoy having you here for a few days of building up more and more collaborations between us. So I'm going to get out of the way. Greg is going to talk to us today about applied cognitive neuroscience, and I think I'll turn it over to you now. Great to have you here, Greg.
Well, thank you all for the invitation. It's great to be back in Switzerland after almost 30 years. Makes me feel really old to say that. But I've had a glorious time in the United States doing a lot of fun science. And my goal here is to talk very broadly about what we've done. I'm going to cover a whole lot of different bases, literally and figuratively. I'll actually talk about baseball a little bit. And I was really inspired by what I saw in the sense. You all really have, you tile a whole lot of different areas of neuroscience and sensory science. And so I wanted to kind of pay an homage instead of staying in one silo of the work we do, but talk about a lot of it. I'm going to try to be really ambitious and get through a lot of slides, so I'll kind of go quick. But stop me at any point in time if you have questions. I'm happy to engage in conversations later. If you get bored by the sports science stuff early, don't worry. We'll switch topics to surgical sciences and psychiatric sciences as we go. So, and get to some of the cool work that Louise is doing towards the end. So, the motivation, how do we fit all of this into one lab, is really behind the diverse skills that humans have. Humans can learn anything. Right? That's the hallmark of humanity, is our neuroplasticity, our ability to acquire skills. And you can see that right here in these different graphs. We learn how to read. We learn how to drive. We forget how to drive. We learn how to juggle. We learn how to play music. Some music is easy. You can become an expert in the kazoo after like a few weeks, but you keep learning and gaining new skills at the piano and the violin after a decade or more. We also can learn how to relearn, right? We have events in our lives, traumas that cause disruption of our plasticity or disruption of our function, and we can relearn after trauma to the brain. We can learn after trauma to the body. We can relearn how to control our mood and regulation. So all of this kind of falls under the inspiration of neuroplasticity. And as a cognitive neuroscientist, at heart, I'm a mathematical psychologist. So what we do is build models. We have great models, kind of explanatory models of different aspects of learning, things from Hebbian plasticity, the fire together, wire together aspects of learning, to reinforcement learning. Feedback drives better learning. the power law of learning. We learn really quickly, early, but more incremental gains later. We have adaptive learning. So, things that are too easy or too hard don't reinforce learning. We learn best in this kind of range of just hard enough. And then a real holy grail of learning is the ability to transfer that learning, right? You learn in the classroom so that you could succeed in your job. You practice in the gym so that you could succeed on the court. And what we really want to do is engender this generalized learning so that what we learn transfers to gains in new arenas. And so the mission of my lab is really to test and develop neuroscience-informed approaches to improve learning and to optimize performance. So this is just a characteristic learning curve. As you practice, your skill goes up. As you stop practicing, your skill goes down. You don't retain everything. And what we want to do is we want to make that curve steeper. You want to learn faster. You want to learn higher. You want to retain better. You don't want to forget as much. But you also want to have people perform in the right context, right? If this kind of Yerkes-Dodson, if you're too aroused or under aroused, you don't perform well. Well, how do you learn to be in the right level of arousement? Also, how do we find the right learners for the job, right? Is this person going to be a great surgeon? Is that person going to be a great baseball player? So these are all questions that we try to tackle in our lab. And on the one side, when we're talking about high performers and expertise, we're talking about peak performance. And when we're talking about recovery and therapeutics, we're talking about rehabilitation. And it may be different in Switzerland, but in the United States, funding really gravitates towards these two ends, right? Our National Institute of Health funds therapeutic research. Our military and other groups fund kind of peak performance. So, over my career, we've gone from a lot of kind of basic science to a lot of applied science, and a lot of the funding falls either studying high performers or studying the kind of medical model of rehabilitation and learning. So this is an outline of the talk. I'm going to give you just a really quick kind of framework of how we do applied cognitive neuroscience, call it our big tent approach. And then I'll go through 3 different applications. I'm actually not going to go so much into future directions. I'll just kind of give you some punchlines at the end. But, Micah is an expert in cognitive neuroscience. He's one of the leaders in the field. And this is what we do, brain and behavior, right? How do those two things work together? How do the mechanisms of brain lead to the processes of behavior? Well, in the behavior side of things, we quantify through psychometric tests or neuropsychological assessments. So we have clinical scales. We try to validate, make them reliable. On the psychometric side, we try to be quantitative. We're talking about sensory processing. We talk about psychophysics, the physical entities of sound and light. They're quantified in kind of a physical sense. So that's the behavior. When we're talking about the brain, well, the nervous system is everywhere. It's all throughout the body. So we do a lot of physiological monitoring, heart rate, eye tracking, things that aren't in the central nervous system. When we're looking at how we could do this in applied sense, we use a lot of simulations. So we'll talk a little bit about some of the simulations we do, particularly with surgery, either using the simulations to do surgery, so like robotic surgery, or simulating the surgical environment so that you can practice in a safe context. When we're talking about, whoa, sorry, I blasted on head real fast. When we're talking about the brain, we measure EEG, the electrical signal of the brain, and we could talk about this in terms of waves and oscillations and the dynamics, because you have high temporal resolution. And when we're talking about functional magnetic resonance imaging, we're talking about the networks, and we're talking about the areas, and also talking about the dynamics. There's a lot of tricks we could play of how to measure the brain in action. The brain's an electrical organ. This is kind of like, after years of doing this, one of my colleagues got up, Holly Listenby, she started to talk with the brain's an electrical organ. I was like, holy cow, it really is an electrical organ. It's just a bunch of batteries in there. Well, as an electrical organ, we can manipulate it through electromagnetism. So while we can measure things on the outside with EEG and MRI and other approaches, we can also causally perturb the brain. And we can do this both for therapeutic benefit, brain's not working right, there's a pathology, we can up or down regulate it using tricks like transcranial electric stimulation, you inject current in, or through magnetic stimulation, where you don't have to worry about this resistance and a shock and things of that nature. And you could drive therapeutic effects. And these are at different places, FDA, federally approved therapeutic approaches. And I'll kind of end the talk talking about some of these, but they're also causal, right? And you're not just measuring A and B as a correlation, you're actually measuring A causes B when you do this. So these are all tools in the tool shed that we use in our Big Tense Neuroscience program. We do basic science, that's kind of the cornerstone, lots of studies with university students. But as I said, that's kind of phased out a lot in favor of applied science. So I'll talk some about some of the peak performance science we do, work with baseball. It's really quantitative sport, so it's really nice for measuring the relationship between vision and performance. I'll talk some about the peak performance with surgery, and then I'll end with some of our work on neuromodulation, so therapeutic interventions. And that's just what I just said. So we were in this, chance favors the person who's, I guess, prepared. We were in the right place, right at this point in time when Nike had gotten it. You think of Nike, you think of shoes. You're wearing awesome, those Air Force Ones. But Nike, for a little while, got into performance. They developed these systems for measuring vision in athletes. Totally not in their wheelhouse. And we were able to kind of start this collaboration with them and get access to big data. My lab is big by lab standard, but Nike is huge. So we were able to really leverage some big data and do data science. This was, again, funded by DARPA, one of the military agencies in the Army Research Office. And it's been pretty profitable for us in terms of our scientific outputs. There's kind of two cornerstones of what we call sports vision or performance vision. And there's this kind of hierarchy. At the bottom, you can think of this really in terms of all vision. There's low-level processes, the receptors, the transducers, the hardware, the monocular processes that might happen in one eye, then the binocular processes that fuse between the eyes, then the mechanics, the kind of integration in the brain of like attention and perception, working memory, and all the things that happen in the software of the brain. And then you have to make this work in with your motor system. And then the output is something you're trying to do on the field when you're talking about sports. So it's the end goal is trying to make this platform as strong as possible and get rid of any bottlenecks. On the other side is, again, that idea of generalized learning, training. So if vision is essential for some sporting activity and you improve that visual ability, do you improve sports performance? And so this is the assessment and the training sides of what we're measuring in sports. So we try to assess what, if any, visual abilities are associated with better performance. And when you do this, like before the season, you're trying to predict, right? It's a predictive biomarker as far as we get in sports science. Or you're trying to diagnose somebody. You might have an athlete comes in, you measure their vision and cognition, then they have a traumatic brain injury, a concussion. And so you can use these baseline scores to diagnose the injury and use it in a return to play sense to say, okay, well, even though you look normal by normal standards, at baseline, you had really amazing abilities. Your working memory and your vision were really good. So maybe you're not ready to go back out. So it's a diagnostic measure. And when we're talking about the interventions, we're really talking about the kind of two things I talked about before: training to improve skills, or recovery to recover from the injury you might have sustained. So just talking briefly about these assessments, these are the systems that were developed. On your left is the original Nike system. On the right is the version once Nike moved out of space, developed by another company called Synaptic. And what was really cool about this, they tried to basically build an optometry office into a light board, right? This kind of digital kiosk. So they had 9 different tasks, actually 10 in the newest version. The first ones were the things you do in an eye doctor's office. Like you measure your acuity, measure contrast sensitivity, measure depth perception. Then the next level was things like your ability to converge and diverge and track. And then it had that big light board. And the next level of questions were eye-hand coordination, visual motor control. And so this was a standardized system. And so what you get back is your performance profile. So with hundreds, thousands, tens of thousands of athletes, you could compare yourself to your peer group. If you're a football, here it calls football, right? If you're a football goalie, you can be compared to other football goalies. And you can also measure yourself over time, before training, before injury, after injury. And it was this really big database. So we got this. It was awesome. It was like we started this collaboration. They started working with us. We did the most boring things in the world, like reading patents. What kind of scientist ever thinks I'm going to read the patents of what a company invented, but you got to do that. Well, we can't trust it. We had to test it. So we did a bunch of laboratory experiments, and we did things like cross-validate it to standard clinical measures, things like a Snellen eye chart, things you would see in an eye doctor's office. And most of them did a pretty good job. Some of them didn't do as good a job. We kind of prioritized our analysis of the things that were reliable, but most of them were. We also looked at how stable they are, what changes, what doesn't. The ones down here, they're too small to see, but you can see the slope of that line. There was learning over multiple practice sessions in things that really involved visual, motor control, and cognition, not so much in things like acuity, right? Your acuity only doesn't change much from day to day. You sit here practice looking at the screen all day, you're not going to suddenly get better acuity. So that was, background, but things got really interesting when we had a data use agreement with them. So now it went from let's take a year to collect 300 people to they send us an Excel file and we have 3,000 people. So now we can do big science, big data on it. And these weren't just any people. These were some of the best athletes in the world. Not both the Nike's stable of contract athletes, but the national teams, the Olympic teams. So we got out some really good chunks of data, including nearly 400 professional baseball players. So we could take that and we can compare that to their baseball performance, right? That's really the peak of that pyramid is not your test scores, but it's how did you do in the games. So we did a few kind of cool early experiments on this data. This was not planned analysis of a null hypothesis test. So we used really exploratory measures like Bayesian statistics. And we did things like look at variability over the whole sample. So what you, I still need to hit that button. There we go. So what you see here is different levels of athletes from high school to college to pro. And as you might expect, in some of these abilities, they got better and better. So eye-hand coordination is better in professional athletes than high school than college. Other things, basically everything got somewhat better at higher levels. But what was most interesting is looking at these kind of cross-sectional differences. So some athletes play sports where you're tracking, right? In football, sorry. You're tracking your teammates and opponents. In other sports, your main goal is to just intercept something. Baseball, cricket, you stand there, it moves. So you could classify sports in terms of the types of demands they place. And lo and behold, we found signatures of these sports in their performance. So athletes who played these tracking sports had better sensitivity. They could pick up cues easily. Players who played sports that were dynamic could track things better. This is a naturalistic sample. This wasn't something that I invented. So it's kind of cool to have this reinforcement of these abilities in the data. Well, great. There's variability, but does it actually predict? Can we measure the base of the pyramid and tell us anything about the peak of the pyramid? So again, we applied these Bayesian statistics. We had 250 professional baseball players that played at least some amount. And they played at different levels. So some of them were in the major leagues, the highest level. Some of them were in the next level down. So I had this great PhD student. He was the kind of person that would just enter data competitions and win. He actually works for Major League Baseball now. He's like the director of analysis for one of the major MLB teams. And he went in and he said, okay, well, we can't just compare across all these levels of sport. Let's normalize. So he found all the players over multiple years who played in multiple levels and then created these normalizations. So you could say, hitting 300%, 30% in major leagues is like hitting 40% in the minor leagues. And so he normalized it. And then we ran these Bayesian models. and basically said, if we take things we already know matter, like what position you play, how old you are, that's our base model. And then we compare it to a model that has the vision assessments. Do the vision assessments add any information? And lo and behold, they did. And they added information for things that related to what we would expect, like how well you hit a ball, not how hard you hit a ball. So we found that there's some evidence here that there's variability. Vision differs across people. Vision differs across people with different experiences. There's a nature and a nurture in here. Do they play these sports because their vision is good, or does their vision get good in a certain way because of sports? We don't know. We can't answer those questions. But we also find that these visual abilities predict their performance. So that was pretty cool. But that's the assessment side. What we really want to do in our applications is actually move the needle, make people better at vision and make people better at playing sports. And so we had this great opportunity to start to use some of these tools. Historically, what athletes and trainers did was things that came out of vision therapy. The things, if you had a patient with amblyopia or strabismus and then their eyes aren't aligning, and that's what they used for decades on how to train baseball players. So write words on a baseball, throw it, and have the person read the words. But all of a sudden, we're in this new digital world, right? You all have some great virtual reality researchers here. Well, we can use that now. So we used a whole bunch of digital tools, and I'll tell you some about what we did with these glasses that shutter open and close. They're called strobe glasses. They're liquid crystal lenses, and you can control how fast they shutter open and close. So this is like creating a resistance for vision. So instead of having full vision, are any runners here? Right, you go run up on the top of one of these Alp mountains where the air is really thin. It's hard. Your blood is not getting the oxygen. You come down to lake level, it's much easier. Well, this is like training under thin vision and then, performing under full vision. So, sorry, I kind of fell behind there. This is just an illustration, and just say, athletes and trainers were trying to do this before these glasses were invented. They'd actually literally have like goalkeepers try to block shots with the strobe lights, big dark room and the strobes going off. There's this great video of Ronaldo. I don't know if y'all have ever seen it, like vision is essential, and you can make you do with less vision and perform at a high level like he does. This is just showing what it looks like. Here you can see through the transparent state, and then it shutters and it's opaque. So this shutters open and close. And basically, whenever it's transparent, it's 100 milliseconds open, and then you can control it and make it go slower and slower by making the opaque state longer and longer. So we had people practice sports doing this. And when I was at Duke, we have a great sports program at that university and a really, impactful basketball program. So we were able to work with them. And we did randomized controlled trials. So we'd have them, one group train under the strobes, another one train with just eyewear that stayed transparent. And they would do stuff like this. They're dribbling basketballs while they're bouncing tennis balls back and forth with the other hand while these glasses are shuttering open and closed. And we had jugglers juggle, and we had hockey players play hockey. So we had, really a whole bunch of different types of training. And we saw, well, does this change vision? And lo and behold, it did. We hypothesized it wouldn't change kind of spatial vision. We thought it would change temporal vision. So we had them do things like measure their sensitivity to motion. Which of these two intervals had the coherent moving dots? And one of them did, and one of them didn't. And before training, it took some percent, 10% to detect it correctly. And after training, both groups got better, but the group that trained with the strobes in the blue got more better in this fovea, but not in the periphery. We found that the useful field of view got better. We had them do a dual task. They had to attend to the center and the periphery. And the group that trained with the strobes got better. The other group didn't. But some things didn't get better. So like multiple object tracking, which took a lot of time, track objects for many seconds, didn't get better. And we weren't the only group doing this. This is a review paper we wrote. It had, I think at this time, about 7 or 8 studies. Now there's probably 20, and not just with athletes. They have some cool studies doing this with surgeons. surgeons training at surgery wearing these glasses that are shuttering open closed, and they get better at not actually live patients, but surgical simulations and drills. So we can move the needle. We can make people's vision better with vision interventions, but can we make their sports performance better?
I'm just wondering how many people were the effect of sight for seconds, given that we use also cycles we use, especially if you play hockey, you can kind of see exactly what the spatial aspect.
I don't think they're I think that's part of, I don't think they're separate, right? You have to use vision in the dynamics. So we were measuring vision outputs, but we were training the whole system. They had to do dynamic vision activities while vision was interrupted. So yes, it also interrupted proprioception, and I think that's kind of the point of doing this in the dynamic space. But we didn't measure the proprioceptive output in our work, but in some of these other studies they did. They measured aspects of, well, actually some of them measured a whole bunch of different things. Some measured EEG, some measured balance. So in our work, we just really looked at vision, but I think that they go together in the training. Cool. So then the next question is like, can we actually make people better at sports, right? That's the end goal. So we were able to do an actual randomized controlled trial, pre-registered with athletes, and measure their output of their sports performance. So we had baseball players from Duke and another school, Indiana, and this was 20 baseball players. They did 10 weeks of training, and one group trained with a combination of different devices, those strobe glasses, but also things like anticipation using a light rail, some other dynamic vision drills, like you have to track things on the screen. And then in the other group, the other half of the people, did drills that were meant to be placebos. And this is hard, right? In drug trials, a placebo is a pill, right? And you don't know which pill you got. But in sports, like these glasses are flashing, right? So what we ended up doing was actually hearkening back to the way the vision therapy studies did their controls, which is to basically have people do binocular drills monocularly, or create some manipulation of vision and then take it away. So they would do things like train on binocular drills with one eye, and you would theorize that wouldn't do anything. You're not improving binocular vision. We also had them do things like Jenga, a game where you like stack blocks, or Simon and things like that. And these are all sitting in the lab, and we play with them sometimes. And we measured several things. We measured their expectations. We measured, did they get better at that sensory station at the beginning? We measured, did they get better at batting in the baseball games the next season? And none of those changed. We didn't have great ability to measure game performance itself, like in the games, because a lot of the players, you had injuries, you had people graduate, you had people leave, you had new people come in. So it was hard to get pre-post and everybody. But we had them do thousands of at-bats of hitting baseballs off a pitching machine. And what we found was the players who trained with the dynamic vision drills, they got better at hitting the balls. You can see here, they hit them higher and they hit them farther. And they hit them farther by about 40 feet. And they hit them higher by about 9 degrees of angle. So this is not game performance, but it's performance. This is hard performance on the field. So it's a step in the right direction. It brings some methods that you don't ever hear about in sports. Randomized controlled trials with athletes, and some vision interventions. So, this is a part of what we do in the lab. It's not all of it. It's a pretty small part right at the moment because we're busy with a lot of other things, but it plays to the idea of vision being important, vision being trainable, vision being something that transfers in applied settings. And it shows things like you can differentiate experts from non-experts. It correlates with your actual performance, correlates with your on-field success. and you could actually train it and improve some. All right, that's sports. Let's jump over here. Another type of athlete are surgeons. Any surgeons in here? Anybody know surgeons? It's super hard, right? It's same idea. You're an expert, you have eye-hand coordination, you have to have a lot of executive control in surgery, right? To be able to pull off surgery. And it's incredibly important. It's even more important than sports. It's necessary. Sometimes it's the only life-saving intervention we have. It's super common in the United States. You know, if you go to the surgeon, they're going to say you should have surgery, almost no matter what it is. And it's really challenging. And so, surgical education is something that unfolds over years and years. And so from our time working with the athletes, the surgeons at Duke and now at UCSD were like, hey, can you do that with our trainees? Can you improve their vision and their performance? So we borrowed a page from the kind of studies we were doing with athletes to see if we could actually improve surgical performance. And this is just to say that, the landscape of how surgical education happens has changed dramatically. It used to be an apprentice trainee model. You would just go and the mentor would say, this is how you do it. And the trainee would watch, and then eventually they would do it. Well, that's changed quite a bit. Now there's a lot more use of simulations. There's a lot more use of kind of planned curriculum. And within the field of surgery, there's a lot more minimally invasive surgery. So it used to be, I had a torn labrum. They would have just cut it all open and fixed it. Well, for me, in this day and age, they make tiny little cuts and insert cameras in there and do laparoscopic or other types of minimally invasive surgery. And that's really hard. It takes a lot of visual motor control to do it because you're operating in a small space. You have things like the fulcrum effect, like when you want to go up, you have to go down. When you want to go right, you have to go left. So there's a lot of learning of visual motor control, and surgeons spend a lot of time practicing and learning. So we asked several questions. I actually don't have time to go through all of them, but two of them that we asked here is, can we improve surgical performance by modulating brain function? And another one we wanted to ask is, can we predict when surgeons are going to make errors? And that's a big, hard question. They're both kind of big, hard questions. So this is an example of laparoscopic surgery. And you can see here's surgeons operating, they inflate the cavity with air, and then you make some small cuts, insert a camera in, you insert your instruments in, and then you're looking up here at a screen ahead of you, and you're operating down here with your hands. Well, you spend a lot of time in residency doing this. Same thing, not in a human, but in a little box. And you spend it doing drills like sutures, moving pegs, cutting shapes, and you get better. There's a learning curve of a first-year resident over multiple practice sessions. So this is like perfect for what we want to do, right? I showed you already, we want to take this learning curve and make it steeper, make it faster. Well, one approach that we use therapeutically, I'll talk a little bit more about later, is the addition of electrical current to the brain. So harkening back to one of my early slides, learning is driven by Hebbian plasticity. Fire together, wire together, right? You have some action. If it's successful, you have some reinforcement by these neurons starting to wire together. Well, the idea behind transcranial direct current stimulation is within this kind of cartoon neuron, you have your baseline threshold or baseline activity. As soon as it crosses a threshold, it fires an action potential, right? That's kind of the way we think about action, neurons function. Well, if you add a little bit of current, shown here in the red, it pushes the baseline closer to that threshold. It makes it easier for that neuron to fire. If you subtract current, it pushes it away, it makes it harder to fire. So if you add current to the right areas, you make it easier to fire, you make it easier to fire together and wire together. And so this is used, somewhat now in kind of post-stroke rehabilitation for like remotor learning. And we wanted to use it here for surgery. So we developed a protocol with that FLS box trainer. Yep.
Because post-stroke has a risk of is there any, is there indication?
Not really in this version, I don't think. I mean, in the research space, you're always very careful. So there may be other, epileptic contraindications that would cause, not to be done. There's many versions of neuromodulation which do have really profound seizure induction concerns, but those are ones where you're firing at a rate. So if you take that same direct current and make it alternating, that can induce epileptic seizures. If we use transcranial magnetic stimulation, it can. The seizure risk, I mean, you are changing the membrane, the thresholds, but it's not profound in a way that causes, I'm actually on a data safety monitoring board for a post-stroke TDCS study, and it's never come up as a safety issue. Do you have a question, Ada? Okay, we'll come back to you later. The long and short of it is that we had people practice a bunch at this. They did 20 minutes, took a break. They did 20 minutes. They did this over 3 sessions. And this is a cartoon version of like the electrodes. We did 2 different montages. We put over the motor cortex in one, and we put over the supplementary motor cortex in another. And then we had a sham condition where we just didn't turn the current on, right? So actually what you do is you turn it on very briefly for a few seconds at the beginning so people feel the tingling, and then you turn it off. So it's off most of the time. And so we tested these different montages. We had a certain amount of current we put in there. This is the consort diagram. So the punchline here is at the end, we had 20 people in each of those 3 groups. The active with 1 montage over the primary motor cortex, the active over the supplementary cortex, and the sham. And it worked. It was like, wow, this worked. Both groups, so the black line is the sham. In both cases, it's just repeated. And the green line is the active in the primary motor cortex. And the orange line is the active in the supplementary cortex. It wasn't statistically significant in the SMA, but it was in the primary motor cortex. So we could actually improve that learning curve. This is, again, like going back to what our goals are, is make learning faster. It actually improved it by 14%, which was equivalent to 2 of the sessions. So we could make the equivalent amount of learning that you need for 6 sessions without the current happen in 4 sessions with the current. And so this was cool. We were like, yeah, that's great. And right at the same time, we published this paper in brain stimulation, and bam, like within like a year, several colleagues had similar findings. We weren't alone. So there was 6 or 7 at the time, and then there was a meta-analysis that came out shortly thereafter that took quantitative view of all of these findings. And this is kind of a meta-analytical plot. It's got your hedges G, so this 0 means there's no effect. If you're over on the left side over here, it means actually it's negative effect. And you can see all of them reported positive effects. The kind of meta-analytic positive effects was modest, and closing in on 1 here. There were different groups. There were different amounts of training. We had the most people and the most training, but also the smallest effects. Everybody else, with fewer people and less training had bigger effects than us, but whatever, we were first. So that was, a cool finding. And so we put in an application. We hope to keep doing this. It was like one of Louisa's first things was submitting this grant. She's like, I didn't know I signed up for this. So let me pivot to what we did with our doing. This is actually ongoing with robotic surgery. Can we predict errors now? Not just can we improve performance, which is again one of the goals, but can we predict when an error is going to happen? And for this purpose, we turned to a robotic surgical system called the Da Vinci. So this has been a really revolutionary, I guess it depends on who you ask, but revolutionary use of human-machine teaming, where the human, the surgeon, is sitting in here looking at a heads-up display with all these different controls of the camera and the instruments. And the back here, you can't see it great, is this robot with like 6 different arms, and you put in, sutures or cauterizers, and it has, digital control and superfine precision. And it is something that surgeons who end up getting board certified in robotic surgery spend a lot of time doing this. They have to practice a lot because there's so many different pieces to it. It's a real complicated piece of equipment. And so this is what we did. Here's one of our surgeons in here. You can see, we're looking at the heads-up display. And we didn't do this with live surgery. We did this with the digital simulations that they do during their training. And some of them are like children's games, like run this little circle over this ring. You might have played this when you're a kid. It's like Simon. If you touch the ring, you get penalized. Other ones were like sutures or things of that nature. And we could measure their behavior really well. We can measure it both through the output of the robot, but also by applying neural net classifiers to the video image. So we get these intervals of, okay, you're doing great, okay, here's an error. All right, and they're error for a second or 3 seconds, however long they were in that error state. And we measured their EEG, that's their EEG, so on their head. And we also measured their heart rate. So we're looking at heart rate variability, and you can see that here. So we actually have 3 papers. The first paper had 19, then we replicated it in a bigger sample, so 57 participants. And we calculated a whole bunch of metrics of the heart rate, basically the periodicity of the heart rate. And you may have heard about heart rate variability, and under this theory that more variability is actually a good thing, right? If you have less variability, you have less arousal and resilience physiologically. And then we did your typical tricks with EEG. We broke it down into power bands, and we ran this through models. And what did we find? Well, this is a scatter plot of the performance, and this is errors versus time to completion. So this is a speed accuracy trade-off right here. So if you're down in this corner, you're making few errors and doing it quickly. If you're in this corner, you're making lots of errors and doing it slowly. You want to be in this corner. We color-coded the years of experience that they had, and you can see that the dots down in this corner tend to be redder. They have more experience training, things you would expect, right? So we could kind of get a good handle on their behavior. But what was really cool is that when we compare the intervals of air to the intervals of non-air, we found differences. And we found differences in their heart rate, as you might expect. As I just said, having higher heart rate variability is good. During the air intervals, they had lower heart rate variability. In the EEG, we found differences in the frequency content. And we tended to find increases in the low frequency and decreases in the high frequency for errors versus non-errors. So this is kind of possibly, if I anthropomorphize it, slower brain activity, less active cognition. What was cool is we applied these machine learning approaches, and we could actually classify with pretty high accuracy the error versus non-error intervals. And we could also classify with really high accuracy, almost 100% accuracy, whether someone was a high performer, whether they're in this kind of bottom corner, or a low performer, if they were in the upper right corner. And one of the tantalizing things is if we actually work backwards in time before the error, we could also predict the occurrence of an error. So with 85% accuracy, and it's about 2 seconds with the EEG, and it's slower. You have to go back about 15 seconds in the heart rate. It's a much slower time constant to measure. But you can actually, above chance, predict upcoming errors. So this is early days. This is kind of a first proof of concept. We have a colleague in Toronto who's doing a similar thing. She's actually videoing real surgery and then having experts rate when a real error happens in real surgery, and they're wearing EEG. The surgeon actually wearing EEG during the surgery, and then they go back and look backwards in time before the error, and they also find something pretty similar. They could find these signatures of changes in the frequency content. So, when I'm trying to sell this to funders, I call it an early warning system we're trying to develop.
I just want to go deeper in details. What does it mean when you can detect 2 seconds before? What is this? Is it error? What is
Yeah.
So it means that person is guilty. I mean, it's an ethical question.
I want to be careful because this is machine learning. You know, we're just identifying patterns, right? And I already wasn't too careful. I already anthropomorphized it some and called it active cognition. And you know, the heart rate stuff usually gets attributed to autonomic arousal, right? So you're not aroused. You're and the same kind of story of like the high to low frequency in the brain. So if I had to like make something up, that's what I would say. But I also want to be careful because this is the first time this has been demonstrated. We're applying machine learning approaches here, and we're just identifying patterns in data. So I'm going to try not to answer your question too well. Okay, so just in the last couple minutes, take care. I'm impressed you made it this long. My kids can't sit through my talks. They can't even sit through dinner with me. Thank you.
Thanks.
You're welcome. All right, so in the last few minutes, I'm actually talking about the kind of some of the coolest stuff we're doing right now, which is now we're going from that peak performers, surgeons, and athletes to patients with depression and mood disorders. And so we do similar tricks. We measure EEG and we try to have predictive or diagnostic biomarkers of, is a patient with depression going to respond? Can you differentiate somebody with unipolar or bipolar depression from their EEG signatures? These are all, same tricks, different ponies, right? We're trying to apply, our methodology in different spaces here. And again, I already mentioned the brain is an electrochemical organ. I'm going to focus on the electrical side of this. And medications target chemical receptors. They alter downstream mechanisms, but they're very diffuse, right? There's not like a cell for depression, right? And there's a lot of side effects. So when you use device-based approaches, you can actually target the pathology a little bit better. You could argue both sides, but since I'm presenting the electromagnetic stuff, I'll say that it has less side effects. There is seizure potential. We try to mitigate that. But you could kind of directly, exogenously change the neural function that we think is pathological. And so you could use approaches like TMS. Micah has a TMS system that's collecting dust in his lab. I'm sure she has to dust it off and use it, but it uses the principles of electromagnetic induction, the right-hand rule of physics. So you pass a current through a coil, and that turns it into a magnetic field. Same thing you have going on with like fMRI. That magnetic field passes through tissue without any resistance, and it causes changes in action potentials. Again, cause is causal. So it changes spiking. And it's not super precise. I didn't mean to say, it's like you could hit an axon or a neuron or even an area. It's pretty diffuse. But it's temporally precise, and it has relatively few side effects. Taking what we've learned from imaging, lots of fMRI, lots of EEG, lots of PET, looking at the psychopathology of mood disorders, we kind of find these circuit level dysregulation. So if you talk about depression in a kind of a circuit sense, we have kind of a hypoactive frontal cortex, which isn't really regulating the emotional centers in the limbic system. Well, TMS can't get deep down to the limbic system, but you certainly can modulate the frontal cortex, things that are more superficial. And there's also been work showing that this connectivity between the frontal cortex and the limbic system, specifically the subgenual nucleus, is pathological. It's dysregulated, and the degree of that pathology, how anti-correlated they are with each other, predicts both how depressed you are and how well you'll respond to antidepressants. So it's kind of the idea that you're regularizing this aberrant pathological network. And so you could do this you could modulate it with different approaches that are kind of rapid acting. So this field of interventional psychiatry kind of has three branches, traditionally true, two: psychotherapy, cognitive behavioral, dialectic, the words are the medicine. Pharmacotherapy, slow-acting drugs that change neurotransmitter reuptake in most cases. So they affect serotonin is the most common one, but there's dopaminergic drugs. And then there's this branch of interventional approaches, things like TMS, electroconvulsive therapy. So actually, we've known for 100 years that inducing a seizure has an antidepressant effect. It also has a bunch of side effects, cognitive and memory. But over the century, we've gotten better at placing the electrodes and shortening the seizure and things like that to make it better. And we're actually now able to induce the seizures in different ways, like with magnets instead of electricity, which also seems to have less cognitive side effects. And TDCS, the direct current, which I told you about, which kind of worked for a while. It might not work. We're trying to determine if it works. But they all are potentially used, and the TMS and the ECT are both FDA approved in the United States. So when I got to UCSD a few years ago, I was in a really fortunate position to have some great colleagues who also were kind of showing up at the same time. And we developed this interventional psychiatry program. So we have a bunch of investigators. Here's some of them, Louise and some other fantastic graduate students. We've actually expanded a whole bunch of facilities, our medical facilities, our research facility in Guava Hall. We got a paint job, but a horrible paint job. I was so disappointed. I was like, they're painting it. It's going to be great. And they painted it like a worse version of what it was. So at least they're not going to tear it down. That's what I'm happy about. Anyhow, we're running a whole bunch of trials. When patients come in, they're not responding. So the interventional approaches are not first line. First line treatments are psychotherapy and pharmacotherapy. But if you failed those, then you become eligible for these other treatments. And we have trials testing them. And the trials that we're testing are really doing kind of one of two things. They're trying to make them more effective by personalizing. We all talk about personalized medicine. Well, we can personalize to the brain. That's the main thing we're trying to do. The other thing is we're trying to predict, trying to measure things at baseline and say, okay, you should get this treatment because you're going to respond to it, and you should not get that treatment based upon your EEG rhythms, your fMRI. So this one trial, there's a picture of Louise that she's been working on, is really targeting anhedonic depression. So this is the negative affect part of depression. And we're using a whole bunch of kind of cool tricks in this study. First of all, we're using a version of TMS, which is called intermittent theta burst. So it's pulses that happen in patterns, 5 hertz and 50 hertz, and it can be delivered in like 3 minutes. So it's pretty quick. And our colleagues at Stanford have recently developed a version of this that instead of doing 1 treatment a day for multiple weeks, you do 10 treatments a day for 5 straight days. So it's super intense. It's called accelerated. And we're doing that. We're also measuring fMRI, so functional magnetic resonance imaging. We're measuring a few things, but in the resting state, just sit there and don't think of anything. And we can measure the rhythms of the brain, and particularly that circuit I talked about before between the frontal cortex and the cingulate. And then we could point the coil at that hot spot that's most anti-correlated between those two. And that is nominally this version of what they developed at Stanford called SAINT, the Stanford Accelerated Intermittent Theta Burst Stimulation, or Intelligent Stimulation. But we also are doing our own personalization. And instead of using generic frequencies, like is done in all the clinics, we're actually measuring their EEG before treatment. And then we're personalizing the frequencies of the stimulation to their frequencies. And so we're specifically looking at the coupling between the frontal and the parietal cortex during a working memory task. So we're looking at what's called phase amplitude coupling. And the idea here is this is an important part of active cognition that's pathological also in depression. They have cognitive side effects. And there were some preliminary studies that showed that this might work in kind of small samples. And we do this really involved study. We measure the heck out of them. They come in, we do these baseline screens, their clinical scales. We measure their EEG. We measure EEG while we ping their brain with TMS. So this is, I'll tell you about this more in just a moment. We measure their effort reward. We measure their fMRI. They wear actigraphy, these digital phenotyping watches. We measure, while we're doing their clinical scales, we video it. And then we use computer vision approaches to try to find features of affect in their face while they're talking about their psychological experiences. Then they get randomized into those 3 arms: regular frequency of stimulation, our personalized frequency of stimulation, and then a sham version. And then we measure everything afterwards, and people who don't respond, we give them more treatment. This is just to say that we've now enrolled 52 patients. The study's not done, so we aren't unblinded yet. So we don't have like the full results. I'm gonna give you a little teaser. And actually the conference I was at, or the group meeting I was at in Helsinki a few days ago, was actually for this big trial. But we've run a ton of sessions, 2,800 treatment sessions in the lab. And considering that none of these people, none of our coordinators, none of our team was there when we started, it was kind of a Herculean effort. So the last thing to say, I know we're running out of time here, is that much like with EEG, when you kind of present a visual stimulus, you could time lock the EEG and get what's called event-related potential, like the peaks that happen, the kind of canonical, this area lights up and that area lights up. You could do the same thing when you measure EEG and ping the brain with a pulse of TMS, and that's called a TMS evoked potential, or TEP. And when you do that, you can look both at that waveform like we do with ERPs, when you're just doing a visual stimulus or auditory stimulus, but you could also localize that down to a place like the subgenual nucleus, right? You can look at how that ping gets propagated down into the cortex or into the subcortex in the subgenual nucleus. And the last kind of data slide is we do this. We do this in all of our patients. We're still blinded. We don't know which arm they're in. And we're using a kind of a secondary outcome measure here of their HAM-D, one of the depressive scales. But what we've started doing is building models that predict treatment response. So we can take the amplitude of these TEP components, put it through a logistic regression, and then calculate a AUC, area under the curve of a receiver operating characteristic curve. And we get a little bit of prediction. We could say, we could predict from the amplitude of the TEP who is going to respond or not. And it's not fantastic. It's AUC of like 0.64, so very modest, but we're still blinded, and we still have work to do here. But this is the direction, one of the directions we want to go. We want to see if our personalization works better. We won't know that until the end when we unblind. But we also want to see if we measure things like EEG, TMS EEG, fMRI beforehand, can we predict who will respond? And so this is a little bit of preliminary evidence Louise can put into her thesis. So I'll just end by saying a few things. So, in the psychiatric space, and I think this is true in a lot of the medical space, what is the future of treatments? One of those is phenotyping, right? Not everybody has the same symptomology, especially in mood disorders. Depression is a combination, it's a cluster of 9, you have to have 5 out of 9 different symptoms. That could lead to 259 different phenotypes of depression. Well, as we've seen, just some of them are anhedonic, some of them are bipolar. You have different kind of clusters. Well, if we look at things like their EEG or their fMRI, which is what our colleague Connor Liston did, you could find that certain clusters have certain brain dynamics. Other clusters have different brain dynamics. That's what he's showing here in these. And that certain clusters, that's what I think he shows not in this graph, but in the paper, certain clusters and certain phenotypes respond better to certain treatments. So we could phenotype and better, parse out symptom heterogeneity and the physiology of it to have a better mapping onto treatments. Next, we can personalize. So instead of just using generic therapies and generic frequencies in our TMS or generic drug doses, you can say, you get this, and we're going to point this to the part of your brain that's most dysregulated. And that's what was done here. This is a retrospective analysis of 4 different studies that looked at how far from that network, that frontal cingulate network, how far the coil was from the treatment, the perfect treatment site where you'd want it to be. And they found that the closer you were to their personalized target, the better they responded. So that's some of the motivation for personalization, and we're trying to personalize in frequency space. That's the study I just told you about. And then the last one is kind of what I just showed you, is prediction. So you measure things before treatment, and does that predict who will respond best to the treatment? And there's some great work by a colleague. We have an R01 under review right now, hopefully it gets funded, where we can actually just go in and do things like scrape electronic medical records. Not even as deep as like their physiology or their EEG or things like that, but basically say, from your profile, from your phenotype, can we predict not just will you respond to this treatment, but which of these 3 available treatments is the best one for you? So we can predict not just for a treatment, but for several. So in conclusion, and I appreciate your patience, I ran over by a few minutes, through neuroscience informed approaches, we can enhance learning. We do it with surgeons, we do it with athletes, and we can optimize performance. We could say, this is the right treatment. If the performance is therapy, we could say this is the personalized treatment for you and it's the right therapy. And so we continue to work along these lines, and we borrow pages from things like, we do clinical trials in medicine all the time. We never do clinical trials in sports. So we started to, borrow pages and learn from the other side of the tent. So the last 2 slides, it takes a village. We have a whole team of like fantastic people we work with. Here are their pictures. It's a great diverse crew, both from the Duke team and from the UCSD team. And we've been very fortunate to have funding from a lot of kind of cool entities, including NIH, the military. We're part of this Welcome Leap program right now that I was just at, and the WUSI Human Performance Alliance. So thank you for your time and your questions and the invitation.